Today I'm going to present the second day for computational systems biology and bioinformatics, which is in continuation. And today I'm going to discuss about big data analysis or multiomics analysis and data. So here it is big data analysis in genomics, epigenomics, and proteomics. And just to go for introduction to omics data. So for example, genomics, epigenomics, proteomics data, and then we will go for its importance, how to integrate multiomics data, and the role of big data in modern biology, diagnostics, and clinical practices. So first thing is types of genomic data, DNA sequencing, RNA sequencing, SMP profiling. So basically we can say microarray, NGS, single cell sequencing. In NGS, these days mostly the people prefer NGS, where we go for RNA-seq data analysis, whole genome sequencing analysis, epigenetics data analysis, such as DNA methylation, transcription factor binding sites enrichment, or chip-seq data analysis, attack-seq data analysis. Then similarly, we have proteomics data analysis, proteomics data also. And in general, the tools are largest number of tools available. For example, what I do, SAM tool. Then there are like a large number of tools are individually also used. And then there are some big software packages. For example, Use Galaxy and Cufflink, GetK, VWA, Star. The applications are ranging in wide range of fields. For example, diagnostic practices, gene discovery, population genetics, and also to predict the genome size of unknown organisms. Then types of epigenomic data, for example, DNA methylation, histone modification, chip-seq, attack-seq. The tools used for epigenomic data analysis are bisulfite sequencing, chip-seq analysis. Applications are in gene regulation, cancer epigenetics. Now in proteomics, types of proteomic data, mass spectrometry, protein-protein interactions tools are similarly a large number of free tools as well as commercial softwares are available. And some of the most common tools are MaxQuant, Proteome Discoverer. Applications are for discovering the biomarkers, drug target prediction or identifications. Challenges in big or multiomics data analysis are data volume, complexity, integration across multiple platforms, and then normalization or standardization and reproducibility. Publicly available data sets and database are Gene Expression Omnibus, GEO, TCGA, CBio portal, mostly for NGS. GEO contains microarray data as well as NGS dataset for genomics and epigenomics. Then NCBI SRA, EBI, EBA, and then Omics, Nephroseq, and there are some other specific databases also for a specific type of analysis, for example, survival analysis. In this case, it is KM plotter, GPR2, Kaggle, Progene B2. Publicly available databases for multiple purposes, for example, to get the reference genomes, gene sequences, and many more things. And they are Ensemble, UCSC, Genome Browser, GeneCode, NCBI, EBI, UniProt. Data integration strategies are, for example, multiomics, data fusion techniques, machine learning approaches, and AI in multiomics, data visualization and interpretation tools. Now I have summarized the multiomics data by adopting the figure from a previous review paper which was published in Nature Review Cancer. And here it has been beautifully summarized for all levels of data. And for example, we could clearly see SNP, copy number variation or copy number alterations, loss of heterozygosity, LOH, genomic rearrangement, rare variants. Then in epigenome, it is DNA methylation, histone modification, chromatin accessibility, transcription factor binding, miRNAs. In transcriptome level, at transcriptome level, gene expression, alteration, splicing, long non-coding RNAs or LNC RNA, small RNA or sRNA. Then at proteomics level, we study protein expression, post-translational modifications such as phosphorylation, dephosphorylation, cytokine array. Then at metabolomics level, it is metabolite profiling in serum, plasma, urine, CSF. Finally, by integrating these different levels of data, we proceed for the prediction of phenotypes in different cases. For example, in case of human diseases, cancer, metabolite syndromes, psychiatric diseases. It would also be interesting to read this review paper for better understanding of the multi-omics data. And then there are some advanced or most relevant part in big data